This video describes a procedure for tracking T-cell receptor signaling events using dual-channel, simultaneous acquisition, total internal reflection fluorescence microscopy, or TIRF microscopy. First, T-cells are transfected with a plasmid encoding GFP-tagged ZAP70. A glass-supported lipid bilayer, which incorporates lipid-anchored peptide MHC complexes, adhesion molecules, and co-stimulatory molecules, is prepared. The ZAP70 transfected T-cells are added to the bilayer. When the T-cell receptors are engaged by peptide MHC complexes, the T-cell receptor-associated CD3 chains are phosphorylated by the Sark family kinase LCK, and the kinase ZAP70 is recruited from the cytoplasm. The T-cell receptor is then fluorescently labeled, and imaging is performed, in which the cells are immersed in medium with lower refractive index than the cover glass and illuminated by a laser beam at an angle that undergoes total internal reflection. This generates an evanescent wave at the glass-water interface, which decays exponentially into the medium, allowing a penetration depth of 100 to 200 nanometers. Thus, only fluorophores in the plasma membrane, or very close to it, are excited. Analysis of the resulting images reveals signaling takes place in T-cell receptor microclusters.
The main advantage of TIRF microscopy over conventional confocal microscopy is that you get better resolution such that signaling clusters or signaling complexes such as TCR microclusters are easily visualized. Travis Kreitz and Lirong Chen, both postdocs in my lab, will demonstrate how to transfect primary mouse T cells